I'm Laura. I have recently graduated from a PhD degree at the University of Zurich. My research was in breast cancer, specifically studying tumor microenvironment. And what really drew me to that research was actually the method used, which is imaging mass cytometry. And what was attractive for me about the method was that it's very computationally challenging method. And that was the main reason why I started research, but it was great to find out that it's in breast cancer, which is the most common cancer among women. So it was great to also work on a field that affects so many women. 3D imaging mass cytometry is an extension of imaging mass cytometry. And imaging mass cytometry is a method that's analogous to immunofluorescence. So similarly to immunofluorescence, this method uses antibodies to target proteins. If immunofluorescence uses a handful of targets, imaging mass cytometry enables to use up to 40 targets. And this is enabled by metal tag antibodies. For imaging mass cytometry, the tissue is stained with those metal tag antibodies. And then the tissue is ablated by laser to then capture the counts of those metals in each tissue spot. And then these intensities translate into an image. 3D imaging mass cytometry is a workflow that I worked for during my PhD. And this extends then the imaging to third dimension by doing series of 2D images from tissue sections, and then computationally putting it together into a 3D reconstructed model. Thanks to its multiplexity, by having up to 40 different targets, this method really enables to capture the different cell types in a tissue. For example, different immune cell types, but also different state of tumor cells. For example, are the cells dividing fast, or are they dying, or different metabolic state of these tissues. And the important thing is that we can get the information of the different cell types together with the spatial information. So we can really study how biology happens in the tissues. The main benefits of imaging mass cytometry really are the high multiplexity, that we can target 40 proteins simultaneously, and the spatial information that we can get. So this really can provide great insights into tissue biology. I think one of the biggest advances in recent times have been in microfluidics. So that has really enabled to separate individual single cells and in a high throughput manner, and then to do genome sequencing and transcriptome sequencing. This gives massive amount of information about the cells, and there has been also attempts, or actually more like advances in recent times, to also combine this with protein level imaging methods. So combining the multiplex imaging methods together with single cell data coming from genome and transcriptome, and that building up this huge information of tissues, so we could learn about biology really in depth. I'm very hopeful about the future of cancer research because I'm coming from a field of breast cancer, and breast cancer is very well studied cancer type because of the huge amount of funding that has gone into breast cancer research. And I think it really illustrates well what can be achieved if a disease type is funded with such a high level. There has been incredible advancements in personalized therapies, in prevention of the disease, in screening methods, all thanks to the research. So I hope that also other tumor types will have such a funding and will have similar success in the treatment. I had a great PhD experience. I worked with fantastic people. I really enjoyed it. I had an exciting project. Thanks to my PhD as well, I had a chance to work in a big scientific consortium that enabled to work together with great scientists across the world, but not only scientists, but also game designers and animators, because the project was the whole big consortium project also involved developing a virtual reality software for analyzing single cell data. And that was done by game designers and animators. And I think this exposure really showed me what is possible, where I could use my technical and scientific skills together with communication skills, teamwork skills. So now I've transitioned into industry, and I work in a digital health startup in the field of chronic cough. And this startup combines technology, software development, and machine learning to really improve lives of chronic cough people. So I'm really excited about this work, and yeah, this is the challenge for me for upcoming years.